So thanks very much. I know, 11 some years ago, I won the Clark Prize, and I think it's such an important, award, and I've been honored to be a part of this over the last 10 years and see how much it has really advanced the public's awareness of the importance of water and the importance of the people that work in this field. So it's just been fantastic. And congratulations, Rhodes. So I'm a public health water microbiologist. I'm at Michigan State University, and I'm going to be chairing your session. And I think David set the stage very, very well with a challenge, and we're going to have 3 speakers who are going to talk about the different aspects of that challenge. Because really, it's a coupling, it's a merger of the advances made at a university in technology, those advances, the novel experiments, the research, and we're going to hear some of that. The exploration of science, of our big picture world, the coupling of natural systems with the human system now, the world we live in the 21st century. And finally, the translation of that science into management, into decisions, in our water environment that's for the public good. And we're going to have 3 speakers that are going to address both the technology and the novel technology at the science, and then this translational science. And so I'm very pleased that we've got these 3 wonderful individuals in this session. So let's get started with our first speaker, and that's Pedro Alvarez. Pedro holds the George R. Brown Professor of Engineering at Rice University in Houston. He's actually the Clark Prize won the Clark Prize in 2012 for his global leadership and contributions to enhancing water resources sustainability through water pollution control. He's been focusing on new technologies for quite some time, and he's looking at 2 areas, bioremediation and nanotechnology. He really began his career in the 1980s, and his papers are widely cited. He's an international expert. So Pedro, please join us and tell us all about nanotechnology.
Well, thank you. Well, thank you very much. While he finds my presentation, I just want to say that this is a real privilege for me to see in the audience so many friends and so many legends and so many young people just getting ready to pick up the torch. But I'm going to talk to you a little bit about microbial contamination of both drinking water and distribution systems, which, as you know, is still a big problem. 1/2 of all hospital beds in the world are right now being occupied by someone who got sick by a waterborne infectious disease. And the need, I guess, for more selective approaches that cause less collateral damage associated with disinfection by products. Disinfection by products cannot be overstated. So what I want to do today is present a vision of how nanotechnology can help in this regard, and perhaps offer several examples at the proof of concept stage to illustrate what might be down the pipeline in terms of offering alternative approaches to microbial control and disinfection. Maybe a good way to start is to remind us about the importance of microbial control. As you know, I think one of the most important contributions to engineering of the 20th century was disinfection and sanitation, which increased the life of Americans from 47 years old in 1900 to, I think we're about 78 right now. And this reminds us that we can really save more lives than doctors by treating water. But of course, it's very difficult to get rid of microbes completely. They never really go away. And in the United States right now, biofouling and microbial-induced corrosion is a $500 million problem, about 3.1% of our gross national product. If all of these activities associated with microbial control and disinfection were centralized by a single company, that company would be bigger than Apple or than Exxon, to give you an idea, which are the largest companies in the world. And it is very difficult to get rid of biofilms once they form, because they give microbes a lot of competitive advantages. For example, they Food tends to accumulate on surfaces, so they don't have to waste energy swimming or chasing after it. And by being close together, that enables to easier respond genetically to evolutionary pressures to exchange genes more easily and adapt. And therefore, they're very, very resistant to predation and to disinfection. And frankly, the traditional approaches that we have for biofilm control just cannot keep up with the increased complexity and the renewed relevance of this problem. And this is where nanotechnology can sometimes offer some hope for meaningful disruptive innovation. So what is nanotechnology? We all know that the prefix Greek, nano, is a Greek prefix for dwarf. In Spanish, in Italian, and in Portuguese, the word for dwarf is enano. Right, and but this definition, which there are many, this is the one from the National Nanotechnology Initiative, has two important aspects. One is that we're dealing with something very small. For example, this is a human hair, and this is a nanofiber that can be used to transmit electricity or information, or perhaps even to prevent crack propagation in concrete, actually. And the other aspect is that we're manipulating matter at the atomic level to create novel structures with novel, perhaps extraordinary properties. For example, at this scale, gold, which is inert, becomes catalytic. And materials that are insulators, like carbon, become conducting. And things that are opaque, like titanium dioxide, become transparent. So things change. And we can sometimes use these extraordinary properties of nanomaterials to do many different things. For example, at Rice University, Chilling Ling is collaborating with Joan, Lo, and I to try to develop these nano patterns consisting of superhydrophobic dense structures that reject or repel colloids or bacteria from attaching, kind of like the taro root or the lotus leaf root or the shark skin. Or you can also interrupt quorum sensing. I'll tell you a little bit about that in a later. We can also incorporate antimicrobial nanomaterials. I've been using nanosilver along with many, many others like Mark and so on. Many has been actually using copper for this purpose, which also has antibacterial activities, and a more relaxed MCL, 1 milligram per liter as opposed to 0.1 for silver. Or you can use photocatalytic nanomaterials that may endow with a surface with self-cleaning properties. So these are just some examples, and I want to give you a few of them. Let's talk about membranes, which, as you know, are very, very important component of futuristic or advanced water treatment processes. I'm going to focus on micro and ultrafiltration membranes, which are the ones that are more broadly used. They don't need as much pressure, and they give a lot more water. One problem with most membranes is that they're prone to fouling. And of course, that increases the energy requirements and decreases their economic life. So one approach we tried a while ago was to incorporate antimicrobial nanomaterials as we were fabricating membranes. So we added about 1% by dry weight of nanosilver, which was added into a polysulfone membrane for microfiltration, and proceeded to see if it was effective in preventing fouling. And what we did was that we took a membrane and filtered bacteria on top of it on purpose, and then we laid that on top of an agar plate that has full. The nutrients diffuse through the membrane, and if the bacteria are alive, they will grow and they will form colonies that you can see with your naked eye, and you can count them. And if they're dead, they're not going to grow. So we saw that the bacteria, the membrane that was impregnated with silver, was very effective at preventing growth, but also it kind of exerted some kind of negative chemotaxis that discourage the bacteria from coming close to the membrane and attaching. So in that regard, it was working well. And the nice surprise is that the size of a membrane is like this. The size of a virus might be like that. They go right through, at least these microfiltration membranes. But silver releases silver ions, which are the actual critical effector of antimicrobial activity. And they're like missiles that are being shot at the virus. And the bottom line is that the membrane that had silver was not getting any virus through. It had antiviral properties, which was a nice collateral surprise. And there are many other approaches that I don't have time to tell you, but as I told you, both Benny and Mark have added carbon nanotubes to make sure that the microbes die upon direct contact. We can put on the permeate side or on the casing nanophoto-catalysts to, again, have some self-cleaning properties. There are other things that you can release here. You can release D-amino acids, for example, that were discovered last year in Harvard to detach, how should I say, they promote detachment of biofilm. So there is a lot of new agents and approaches here that we can incorporate into so-called responsive membranes that release things when they sense microbial contamination. The second example I want to tell you is about interrupting quorum sensing. So what is quorum sensing? Well, bacteria talk to each other. They do this through chemical signals, for example, homoserine lactone. And the way they talk is that they excrete these molecules, and when the bacteria are crowded and form a quorum, the concentration of these chemical signals reaches a gradient that favors re-entry into the other cells. And when these autoinducers of homoserine lactones go back in, that triggers a series of reactions of biochemical mechanisms that regulate many things like bioluminescence, sporulation, and biofilm formation. So in other words, when bacteria reach this critical mass, this quorum, they tell each other, hey, let's go and attach. Let's form a biofilm. Now, let me tell you how I got, there are 4 ways that you can prevent that. The idea here, the premise is if you stop quorum sensing, you interrupt it, they will not attach. And to block it, you can block the production of the autoinducers by repressing the gene at the transcription level. In other words, nip it right at the butt. Or if the autoinducer or that signal is produced, then destroy the signal. Or use a fake signal produced by another organism as an analog signal that will bind the receptor and inactivate the receptor, or damage the receptor. But these 2, 3, and 4 strategies are very species-specific and are not broadly applicable. So we target this approach, which seems to be more effective, which again is, get it at the root of the problem. Let me tell you very quickly how I got involved in this. I had a student, a doctoral student from NASA, Letty Vega is her name, and she tells me one day, we gotta bring back this heat exchanger from the space station. It just has microbial contamination. Well, how do you know? Well, then the carbon dioxide went up, the pH is down, we're detecting metals that are being dissolved. It's probably foul, and this is important because, as you know, biofouling hinders heat exchange. So they say, we're going to bring it back. So they bring it back and use all kinds of sophisticated microscopic analysis, Auger microscopy, epifluorescent microscopy, and find no biofilm, and they find no evidence of corrosion. Yet they had a lot of microbes that were free swimming in hog's heaven, about 10 to the 6 cells per mil. And in nature, 99% of the microbes are going to be attached, but they weren't attached here. So that was a big puzzle. So we hypothesized that there's got to be something in that liquid that is not killing the microbe because it's at sublethal levels, but yet is preventing them from attaching. And it turned out to be nickel and other divalent ions. The way we figured that out is, for example, we ran some screening tests with this bacterium, Chromobacterium violation, which turns violet whenever it is triggered by quorum sensing. For example, this is a positive control. This is a negative control. This bacteria has been co-incubated with Pseudomonas aeruginosa, which produces a signal that is an analog that blocks the receptor but doesn't induce quorum sensing. But anyways, over here we're increasing the nickel concentration, and there's a level at which nickel stops quorum sensing without killing the bacteria. So we use many other strains to see if that could be reproduced in other bacteria. In this example, we see Burkholderia cepacia. And what we're seeing here is that the planktonic viability is not affected. These bacteria are alive. nickel is not killing them. Nickel is not even regulated in drinking water, as you know. There's not even a secondary drinking water standard for nickel. And however, the ones that are forming biofilm, as you increase the concentration of nickel, the amount of biofilm decreases. Then, to establish a clear etiology here, we add back the signal, the chemical signal that you can isolate, the homoserine lactone, and then boom, the biofilm formation goes back up. So that shows that that clearly has to do with repressing the production of that signal. And to make sure that that was occurring, well, so the implication is if you can get bacteria from expressing their quorum sensing signals, you will get them to stop from forming biofilm. So we went ahead and tried to see if this was occurring at the transcription level, if we were blocking this process at the transcription level as hypothesized. And this bacteria, conveniently, we know what the gene is that codes for quorum sensing, is the CEP gene. And you can see that that gene is downregulated by 5-fold. And when that happens, that coincides with complete biofilm inhibition. Now, why? What's going on? Well, we don't know, to be honest with you. But what we suspect is, from a mechanistic point of view, is that nickel and also cadmium does the job. But cadmium, of course, has an MCL of 5 parts per billion, same as TCE. You don't want to use cadmium unless it was maybe an industrial setting. But they are competitive antagonists to both manganese, calcium, and maybe even zinc. And that does many things. One of them is prevents bridging and adhesion, and it also prevents the action of some RNA and DNA polymerases, and thus that inhibits quorum sensing. But the point is that nanotechnology could offer opportunities to engineer the particles or at the right size or with the right coating so as that you can have a more controlled release, perhaps even smart release, like on-demand release, that they sense a difference and then we're going to release this, so that you never exceed the MCL and you use this release more judiciously. You don't become depleted from this metal so quickly. The next example is going to have to deal with the oil and gas industry, because I live in Houston now, and that's where all the opportunities are. Unfortunately, the federal funding is drying up. And let me just tell you, this is a multi-billion-dollar problem for them. Micros, for example, convert the good quality light oil into bitumen. They clog up wells, and often you have to abandon them. A lot of that has to do with precipitation of minerals like sulfides. They obstruct the flow, and most importantly, they're responsible for souring. Souring refers to the production of hydrogen sulfide, mainly by sulfide-reducing bacteria in situ. And that's a problem because hydrogen sulfide is a corrosive gas, and it's also a toxicant. I heard of people in China that have died near well fields when there's a sudden burst of hydrogen sulfide and they're just asphyxiated. But this costs them a lot of money. Just a company like BP spends $2 billion a year in biocides, for example, just trying to get them. And biocides that are very marginally effective because they don't get to where they need to go, and they are very non-selective. And they also interfere with other things that they add, like, friction reducers and gel enhancers. So anyways, biofouling happens to be one of the least manageable and most impactful problems that they face right now. So how do we improve the selectivity of microbial control there? Well, this is an idea that we came across from Rice, primarily from Mike Wong. Mike Wong was thinking about increasing the selectivity by creating, mesoporous particles, perhaps made of carbon, in this case we say silica, that where you load them up with a biocide, and then you can cover them with a trap, in this case a triazine trap, that is susceptible to nucleophilic attack by hydrogen sulfide. So if you, this is an example of what we call a responsive particle. If you sense the hydrogen sulfide that is present, the trap will fall off, and then the biocide will be released. Of course, I don't know how feasible this is going to be by the large quantities that are needed, but I just wanted to show you what might be, possible in the future. We don't know what the economies of supply and demand of the economies of scales, once they play, what the prices of these nanoparticles are going to be. But something that will be much cheaper right now is, and also much more selective, is to use bacterial virus or phages. I was talking with Joan earlier that there's about 10 to the 30 phages in the world, by far the largest genetic resource on Earth. For every bacterium that you have in water, you have 10 phages, no matter if it's groundwater or the ocean. Yeah, and they do really, really well in that regard. The idea is to isolate and enrich polyvalent phages that self-propagate, they self-replicate, and inoculate or vaccinate reservoir that way. We did some very preliminary tests by isolating phages from activated sludge and testing them on sulfate-reducing bacteria. And you can see that they stop the production of these biogenic minerals that precipitate and the formation. And we think we can do better if we use cocktails of phages, if we mix them, and if we use a higher multiplicity ratio. Here we're using 1 to 10. In nature, the normal ratio is 10 to 1. So we're using 100 times less than in nature here. So there is some promise there. The final very quick example is the opposite. We're going to use nanotechnology to enhance microbial activities as opposed to hinder them. And this has to do with the Macondo well, the BP release in the Gulf of Mexico. Here, we know that the degradation of the heavy hydrocarbons that remain is the rate-limiting step is the first one, the hydroxylation. That makes hydrocarbons more soluble and more bioavailable and more biodegradable. And we use photocatalysts, in this case, food-grade titanium dioxide, the same that you sprinkle on those white donuts. That's not sugar. A lot of that is titanium dioxide. And we sprinkled it on that so as to promote and kickstart bioremediation. And according to our hypothesis, we see that after 1 day exposure to titanium dioxide, the amount of hydrocarbons that are dissolved increases, and so does their biodegradation subsequently. And by the way, this is an approach that could also be very useful for polishing the effluent from wastewater treatment plants that carry all these endocrine disruptors that bypass the treatment. You can easily, if you already have UV disinfection, we can add a photocatalyst to destroy, with a very short contact time, all these endocrine disruptors that are escaping. But anyways, to wrap it up, we have seen that there are many antimicrobial nanoparticles that can enable microbial control in many ways that are perhaps greener, that bypass the concerns of, how should I say, disinfection by pros. But I guess the take-home message I want to leave you with is that, of course, nanotechnology is not going to be a panacea that will be universally applicable. But it is very rapidly earning an important place in the menu from which we select solutions to our microbial control and biofilm eradication challenges. With that, I want to just quickly thank the agencies that funded this work, the students that conducted the experiments and got the data I presented, and of course, thanking you for your attention.
Paper's open for questions. Yeah.
Pedro, some of the alternative disinfectants that you talked about have very high MCLs, but they have very low water quality criteria. For example, nickel 10 to the minus 7 molar, silver 10 to the minus 8, and the triazines maybe even 10 to the minus 9.
Yep.
A couple of years ago, a company tried to release a silver nanoparticle impregnated washing machine in California, and it was essentially banned because of concerns about silver getting out in the environment and having effects in the aquatic ecosystem. So how do we temper this message of enthusiasm for alternative biocides when they have the potential to cause large problems in aquatic ecosystems?
A very, very good point. We certainly need to be always aware of unintended impacts of our solutions. In silver, another thing that I just found out is the impact that it has to activated sludge. It makes the flocks fall apart rather easily. The trick is controlled release in how much you use. You don't want to use too much so that it causes this problem, and it also costs you money. You have economic incentives as well as eco-responsibility. In the case of the silver, that we were putting on the membranes, the amount that was being released was about 10 times below the MCL, which was at 0.1 micrograms per liter. So you have to be extremely wary about putting the right coatings or somehow modulating the release so that it doesn't become a problem. I agree with you. The other alternative is use something less toxic like copper, but it's also less potent as a bactericide.
Other question? So I want to ask you about the bacteriophage. How are you finding the soup? I mean, how are you trying to explore what's the optimal soup of phage that you could use?
Yeah. So the current methods that we have for isolating phages are extremely biased to isolating narrow host phages. What we're doing is we believe that the determinant is absorption. And so those that absorb, that don't, we made them absorb to one bacterium, then to another. Those that absorb go to another one. And so we go through a series of enrichments with different hosts. And after you get through 10, what you end up with is a polyvalent phage that is a little bit slower, but is much more promiscuous, if you wish. But, I think phage technology has tremendous opportunities for detecting pathogens, for more selective disinfection, for more selective control of pests in agriculture, and so on. You have an uphill battle with public relations. I never use the word virus. I always call them phages because otherwise people would be turned off. But I think that that's something that is a very fertile area of research.
All right, let's thank Pedro one more time.